Hello friends, AIIMS Botane next episode. In this episode, the main chapter is biological classification second episode. And what all things we'll be discussing in this episode. For that, I will share the screen so it will be convenient for you. I think screen came. So we can proceed. AIIMS Botane next episode. Meanwhile, short question I'm daily doing. One is in the lunch time around 12:30 to 1:30. Then after that, from 8:30 to 10:30 gap in the night, 2 more. Presently, I am doing second year questions. Gradually, I will also include first year questions. So first year 2 questions, second year 2 questions. Second year, we are proceeding with the genetics. Now we are continuing it. Already one question today itself I put. You can check it in AIIMS Botane. So subscribe it. So what all things we'll be discussing in biological classification second episode. As usual, there will be an introduction. And we'll be continuing monoerens, where archaebacteria, cyanobacteria, mycoplasma. After that, 5 theory question, 5 NEET model question with the key as usual. So we can start with the introduction. We already studied bacteria. So many other microbes are available along with the bacteria, like archaebacteria, cyanobacteria, mycoplasma, all comes in our syllabus. So we'll be continuing with the archaebacteria in biological classification 2. Archaebacteria, archae itself means very ancient. So archaebacteria, very ancient bacteria in that sense. Sometimes in some editions, some books, it is written as oldest of living fossil. That is archaebacteria, oldest of living fossil. Some books, it is written. And they have some special features. They are able to face extreme conditions. And which are conditions or what are their habit and habitat that we can easily get when we pass through their names. So first group is allophiles. Second is thermoacidophiles. Then third is methanogens. Allophile means their habitat will be extreme salty condition. Hallo. Thermoacidophile. So temperature will be more, acidicity will be more. Phyle means love, so hot springs. And methanogens concerned with the marshy area. I think first page is clear, so we can proceed to second page. So when we proceed with the second page, we are starting with the archaebacteria, how it is able to survive in extreme conditions. It starts with the cell wall. Cell wall in archaebacteria is special because it differs from bacterial cell wall that we already studied. It is peptidoglycan in a typical bacteria. Here it is different. In archaebacteria, it has special nature by which these organisms are able to face extreme conditions. And in archaebacteria cell wall that consists of N-acetyl tallose amuronic acid, NAM. That is peculiarity of cell wall, not peptidoglycan. Another chemical NAM. N-acetyl tallose amuronic acid, NAM. I think name is not so familiar to us, NAM. Then comes cell membrane. Cell membrane also some speciality. It consists of branched chain lipids with the phytanyl side chain, which decreases the membrane permeability. So it can adapt. Now comes the term introns. Introns and exons, that is second year genetical topic. Still, genetics will be studying it introns for time being here. Presence of introns. It is not a feature commonly seen in monoerens. Monoerens, we will not normally use introns, but in archaebacteria, it is seen. Normally, archaebacteria is divided into 3 groups, which include methanogens, allophiles, thermoacidophiles. Now we will be discussing these 3 groups. First is methanogen. Gen means production, genesis, microsporogenesis likewise. So meaning here is methanogens, methane producing. And they are commonly seen in marshy areas. They can even produce methane from carbon dioxide, formic acid, methanol. These organisms can be seen in guts of ruminant animals. I think up to that it is clear. So we can proceed to next page. This is the reason why we are able to produce biogas from dung of cattle, because that microbe is there. Methanogens are chemoautotrophs they are obligate, strict anaerobes. And typical example is methanobacterium and methanococcus. This aspect, how that methane gas or biogas is produced, we'll be studying in second year under sewage treatment plant, STP. Now comes second group, that is halophiles. One meaning of halo is salt. Phily, we are familiar now. Hydrophily, hydrophobic, we study in cell membrane. So phily means love. Does it mean salt-loving? In this context, halophiles. The microbes normally lives in extreme salt. Salt means saline conditions under strong light. When plenty of light is available, they develop a membrane. It is pigmented and also called purple membrane. This is a frequent question. Purple membrane is seen in halophiles. It has pigment. And what the pigment is, commonly what we believe is bacteriorhodopsin. Purpose is to receive sunlight for production of ATP. But remember, this ATP cannot be used for food production. So that indirectly means halophiles will be heterotrophs. Example, Halococcus, halobacterium. I think idea clear. Now we can proceed to third group, that is thermoacidophiles. Thermoacidophiles means thermotemperature acid. Acido means acid. Phyle means love. So in totality here it means these are the organisms that can survive in high temperature and acidicity. See the extreme conditions. And they are living in hot water and acidic conditions. Approximate temperature will be around 80 degrees centigrade. And pH is around 2. Extreme pH 2. And they are chemosynthetic. Under aerobic condition, they can be linked with the sulfuric acid. Under anaerobic condition, they can be linked with H2S production. And they are facultative anaerobes. Example is thermoproteus, thermoplasma. I think up to that it is clear. Three groups. Now comes eubacteria, otherwise true bacteria. And there are thousands of different eubacteria, true bacteria, characterized by the presence of rigid cell wall. And if motile flagellum seen, we will be studying cyanobacteria and mycoplasma after this. So we'll be proceeding to next group, cyanobacteria. Cyanobacteria is commonly called blue-green algae. Short form is BGA. Another name is Cyanophyceae. Another name is Myxophyceae. And normally, scientists believe it is gram-negative. Some exceptions may be there. Normally, gram-negative. Photosynthetic, prokaryotic organism. And they perform oxygenic photosynthesis, chlorophyll A is seen. They are most self-dependent organism. Reason include photosynthesis they can do, nitrogen fixation they can do. They have diverse habitats, and they are early colonizers in succession. Next is habitat and habit. They are mainly aquatic, preferably freshwater, sometimes marine, and some are terrestrial also. Some blue-green algae forms symbiotic association with the different organisms. It can be seen with the angiosperm, gymnosperm, pteridophyte, bryophyte, green algae, fungi, and various organisms in animal side also. Associations. Now, some important association in plant side includes a biofertilizer called Azolla, cyanobacteria's link. That is why it is able to fix atmospheric nitrogen. Cycus, a gymnosperm. Then in animal side, with the protozoa also it will be linked. So protozoa it is endozoically. And that association between these two groups is called cyanella, important question. Now structure and organization. They can be unicellular, colonial, and filamentous. In filamentous, one or more strands will be there called as trichomes, surrounded by mucilage sheath, as shown in the diagram. In BGA, flagella is absent throughout life. Now metabolic activity. Nitrogen fixation is an anaerobic event. The structure linked with it is called heterocyst. Seen in organisms like nostoc, you can find the NCERT diagram given here. Nostoc. Cell walls are thickened, so oxygen cannot enter. So anaerobic condition is provided. So oxygen cannot enter, thus an anaerobic condition is provided. Now photosystem 2 is absent. That time being you may not be doing it is in photosynthesis. So water splitting will not be there. So photosystem 2 is absent in heterocyst. Thus, carbon dioxide fixation will take place in vegetative cells. PS1 is active in heterocyst. PS1 is active in heterocyst. PS2 is absent in heterocyst. PS2 absent, PS1 present. Since PS1 is there, they can generate ATP, and that ATP can be used to fix nitrogen. I think idea clear. Now we can proceed to blue-green algae reproduction. Mode of reproduction in blue green algae is asexual, which include binary fission. Then there is fragmentation, heterocyst. It can develop under certain condition, achainites, hormogonia. Exam point of view, most common terms they are frequently asking. One is binary fission, other is hormogonia. So hormogonia I expanded here. See, filaments, there we can find, see this area that lost the Whittaker turgidity will be lost. So that become biconcave and gradually dead. And then it is called necroidia. So this is necroidal area. Above it and below it, that they are called hormogonia. And they can develop later into new filaments, hormogonia. First hormogonia, you can find O is missing here. Here it is, right? So here O is missing, hormogonia, hormogonia. Same mode of reproduction in cyanobacteria. Necroidia formation, biconcave dead cell, then above below hormogonias separate and develop into new filaments. Economic aspect of BGA, plus and minus. See, in evolution, they are important since they are very ancient organisms. In which context important? Ancient organism, no doubt, able to show, next is important, able to show oxygenic photosynthesis connected with the aerobic form of life and their evolution. It's an important question. They are biofertilizers. They can increase soil fertility, nitrogen content. Example is Tolipothrix and Allosera in rice field. And non-symbiotically also they can work. Ecological succession, in initial part they will be playing a role. In reclamation of soil, user soil, USAR, that means user soil means alkalinity and salinity will be more. So such soils not good for agriculture, converted into useful soil by treating with the BGA. Edible purpose, SCP, for humans and animals. Typical example is spirulina. It's edible, non-toxic, quick growing, packed with vitamin, minerals, antioxidants, and proteins. And many are symbiotic, helps in connection with the minerals like nitrogen, etc. In certain cyanobacteria, certain antibiotics are produced. That is, typical example is lynch pea. And the counter side is they also produce algal blooms, toxin production causing trouble to humans and other animals and life. Example is microcystis. Now, next is small part, that is heterocyst. So when we come to heterocyst, diagram given there, labeling question NCERT, they are specialized nitrogen fixing cells. As in nostoc, connected with mainly nitrogen fixation, rarely that may be connected with the reproduction also. Now a term, not in NCERT, but it's a famous term, Guide-Co phenomenon. So what is Guide-Co phenomenon? It is seen in cyanobacteria. That is complementary chromatic adaptation and enhancing photosynthetic efficiency, Guide-Co phenomenon. Seen in blue-green algae, not in all, some members. Now comes next group, mycoplasma. So when we come to mycoplasma, it has got synonyms like molecules, PPLO, pleuropneumonia-like organism, Jokkers of plant kingdom, MLO, mycoplasma-like organism, MLO. And they are pleomorphic. Pleo, that's an important question. Pleo means not constant when it comes as a prefix. So pleomorphic, shape is not constant, means pleomorphic totality means shape is not constant. In genetics, we'll be studying pleiotropism. Interestingly, cell wall is absent. That is why. One reason why they are pleomorphic. Cell membrane is the outer layer. It is 3-layered. During culture, mycoplasma pollinase looks like fried eggs. Normally non-motile. Certain cases, gliding nature has been seen. Ribosomes will be 70S. Main cell organs, nucleus, all are absent. Now comes mycoplasma diagram. It is labeled, everything given there. They have both the DNA and RNA. RNA is single stranded, DNA will be normally double stranded, ribosomes present. And mycoplasma and antibiotics. See, since cell wall is absent, Jokkers of plant kingdom, wall-less bacteria, the antibiotics which works on the cell wall is ineffective in mycoplasma because that antibiotics works on cell wall. Here it is absent in mycoplasma. Typical example is penicillin. So no cell wall, so penicillin will be ineffective for mycoplasma. Thus, antibiotics works on some other areas, or the mechanism must be utilized against mycoplasma. Typical example is chloramphenicol, streptomycin, erythromycin, etc. Their action is linked with the metabolism. Now, mycoplasma reproduction respiration. Mycoplasma is reproduced by a structure called elementary bodies. Elementary bodies, and mostly they are facultative anaerobes. And there are some important diseases caused by mycoplasma. Once in a while, they're asking for entrance. In plants, the general symptoms almost similar to viruses: stunting, leaf yellowing, witch's brew, phyllodes. Then animals, pneumonia, arthritis, mastitis, reproductive diseases. In humans, again, pneumonia, reproductive diseases, urethritis. And Mycoplasma gallisepticum is one of the smallest bacteria, Mycoplasma gallisepticum. Now, cyanobacteria, two terms. One is called centroplasm, other is called chromoplasm. Centroplasm means central, colorless. That is why it is called centroplasm, having chromatin material. Chromoplasm means peripheral protoplasm, have color due to pigment. And after that, 5 theory question, 5 NEET model question with the key. What all things we learned in this episode? So we can just rewind it. So today we started biology classification, second part. And mainly there is an introduction. Then monorails we continue. And first archaebacteria, what are the features, what are their names? Then what are the specific features? Cell wall, cell membrane, etc. Then after archaebacteria, we started with one by one methanogens. Why the name? What are the products? Then halophiles, again, why the name? What is the characters, etc. Then thermoacidophiles, temperature, acid, etc. How they are able to survive, temperature, pH, etc. Then eubacteria, under with cyanobacteria, different names for cyanobacteria, and oxygenic photosynthesis, habitat and habit. Then associations, including cyanella. Then structure and organization, unicellular colonial filamentous trichomes with the mucilage, flagella absent. Then metabolic activity, heterocyst. Then PS2 absent in heterocyst, PS1 is present. By which they can generate ATP to fix nitrogen, not for consumption, to fix nitrogen. BGA, we continued with the reproduction, asexual only. Then binary fission, fragmentation, heterocyst like kinites, hormogonia. Then hormogonia, what it is, necrodia, what it is, economic aspect of BGA, plus and minus. Then after that, we entered into heterocyst. What is the speciality? Gadeco phenomenon, mycoplasma, synonyms, molecules, PPLO, Joker's, Pancreas, MLO, cell wall is absent. Then cell membrane 3-layered. Then culture media appears like 5X, 70S type ribosome. Other cell organs, including nucleus, absent. Then a diagram of mycoplasma. Then antibiotics, which all will be working, which all will not working. What is the reason? Then mycoplasma reproduction, how it is done. Then diseases caused. Then cyanobacteria, centroplasm, chromoplasm, what are the differences? Then after that, 5 theory question, 5 NEET model question as usual with the key. If you like this episode, subscribe or like it, share it, comment on it. We can meet later. We'll be continuing bio classification because all of the rest of the chapter is over, second year. Now only remaining chapter this one. So we'll be continuing bio classification in next episode. So take care. Bye. Thank you. We can meet later.